Hello friends, I Pratibha Sharma, PGT Biology of Kendra Vidyalaya Junagadh. I am again before you and in continuation to my previous lesson, today we'll start with the second part. Now first, let's recapitulate what we studied in our previous lesson. We studied that flower is the site of sexual reproduction. It consists of essential and non-essential floral wholes. Okay, now before fertilization, in essential floral whole, androecium is the male reproductive structure which is made up of stamen. We studied the parts of stamen, that is anther and filament. Inside the anther, 4 microsporangia are there, and these microsporangia, each microsporangia is covered with 4 wall layers. Okay, outer 3 wall layers, epidermis, endothelium and middle layer, are meant for protection and helps in dehiscence of the anther. Inner to that, the next layer is tapetum. Its function is to give nourishment to the developing pollen grain. So if this tapetum doesn't function properly or it is malfunctioning, the pollen grains which will form in the center, they won't be viable, means they won't be able to germinate. Okay, so this all we studied in previous lesson. Now, today we'll start with the topic microsporogenesis. Here, genesis means synthesis or formation. And what is forming over here? Microspore. So, microsporogenesis, it is the process of formation of microspore from pollen mother cell or microspore mother cell. Okay, so inside the microsporangia, in each microsporangia, there is present sporogenous tissue, as we studied earlier. Each cell of the sporogenous tissue is known as microspore mother cell or pollen mother cell. The process by which these pollen mother cells are forming microspore is known as microsporogenesis. Okay, so you can see in the picture, this is a single microspore mother cell. It is diploid. So it undergoes meiosis. After meiosis 1, it forms microspore dyad, 2 microspores. They further divide by meiosis 2 and results in the formation of tetrad of microspore. So one microspore mother cell, it gives rise to 4 microspores. And since the division here is meiosis, so all the microspores, they are haploid in nature. Whereas microspore mother cell is diploid. So one diploid microspore mother cell give rise to 4 haploid microspores. Okay, now as microspore continues their development, they mature and forms the pollen grain. Initially, they are associated with each other. Later on, as they continue the development, they dissociate from each other, and each single microspore, it develops into a mature pollen grain. In exam, you get question on microsporogenesis. What is microsporogenesis, or how microspores are formed from the microspore mother cell? So you have to explain only up to here. But if it is asked like how a microspore mother cell develops into mature pollen grain, so you have to first explain microsporogenesis, that is these stages, and after that you will explain how a single microspore matures into a pollen grain. Okay, so two ways the questions can be asked. Now, next is stages of microspore maturing into pollen grain. So you can see a single microspore is there. Its nucleus is haploid, that is N, one set of chromosome is there. First, this microspore, it enlarges. Okay, its volume increases. Once the volume increases, formation of vacuole takes place inside it. Due to the formation of large vacuole, the nucleus which was present in the center, it is pushed towards the microspore wall. Later, so you can see a microspore, it enlarges, it expands, and its volume increases, and then nucleus is pushed towards the periphery, and then the nucleus undergoes, microspore nucleus undergoes mitosis, and it results in the formation of two cells inside the microspore. Okay, so initially the nucleus will divide, then the whole cytoplasm divides, and inside the same microspore, two cells are formed. This two-celled structure is known as pollen grain. Okay, so the two cells which are formed are vegetative cell and generative cell. Vegetative cell, it is the larger one, it is bigger in size, and it is rich in food reserves. Lot of food reserve is present in it, and it is having a large irregularly shaped nucleus. On the other hand, the second cell is generative cell, and it is smaller in size. It is spindle in shape. Spindle means pointed at the edges and thick in the middle, and it consists of spindle shaped nucleus. Now this generative cell, it separates from the microspore wall and it is engulfed into the vegetative cell. So in a mature pollen grain, we find that the generative cell, it floats in the cytoplasm of the vegetative cell. Okay. These are all the stages which I discussed. Now, in 60% of angiosperm, pollen grains are shed at this two-cell stage, that is vegetative and generative cell stage. While in 40% of the angiosperm, the generative cell nucleus which is there, it undergoes second mitosis, and it after completing that mitosis, it results in the formation of two male gametes, which are also known as sperm cells. Okay, so now it becomes, you can see this is the vegetative cell, and since the generative cell was floating in the cytoplasm of the vegetative cell, so when it divides and forms the male gamete, that male gametes also floats in the cytoplasm of the generative cell. So this is three-celled stage of pollen grain. Okay, so in 60% of the angiosperm, pollens are released at two-celled stage. This one, vegetative and generative. Whereas in rest 40% of the angiosperm, the pollen grains are released at three-celled stage. Okay, now in 60% where it is released at two-celled stage, once the pollen grain germinates on the stigma and pollen tube formation occurs, at that particular time the generative cell divides and forms the two male gamete. Means finally this also changes into three-celled stage. So that is about microsporogenesis and how microspores matures into pollen grain. Now we'll study the structure of pollen grain. So in a pollen grain, we find that it consists of two cells we already studied. The larger one is vegetative cell. This larger one, this is the vegetative cell, and inner to that is the generative cell, which is spindle shaped. Okay, a pollen grain, they represent the male gametophyte because they carry male gamete either after germination or before. The pollen grains of different species, they vary in shape, they vary in shape and size. Their size varies from 25 to 50 micrometer. Okay, now each pollen grain is covered with two walls. The outer wall layer is known as exine. This exine is hard and it is made up of sporopollenin. Sporopollenin is the most resistant organic material known on Earth. It can withstand high temperature, strong acid, alkali, and there are no enzymes which can degrade it. Okay, so it is highly resistant material. Due to the presence of sporopollenin in their outer wall, they can be well preserved as fossil. This question also comes in exam, why sporopollenins can be preserved as fossils. Okay, so you have to write the one which is there in the bracket. Now in exine, you can see the exine is not thick all over. At certain points, the sporopollenin is absent. These regions where the sporopollenin is absent is known as germ pore. So it is these germ pore. Here in the diagram only one is shown. There are more, okay, at different intervals. Now, when a pollen grain germinates, its inner wall layer, that is intine, it comes out through the germ pore and forms the pollen tube. Okay, so since sporopollenin is not there, it is the thin region of the exine through which the germination of pollen grain is taking place. Now, the inner wall layer is known as intine and it is made up of cellulose and pectin. And a mature pollen grain contains two cells, the vegetative cell and generative cell. In exam, you may get question like write difference between the two cells present in the mature pollen grain. Okay, so you should know the difference between vegetative cell and generative cell. So vegetative cell bigger in size, generative is smaller in size. Vegetative cell contains abundant food reserve, okay, whereas here the food reserve is not there, and the generative cell floats in the cytoplasm of vegetative cell. Nucleus of vegetative cell is large and irregularly shaped, whereas the nucleus of generative cell is spindle shaped. And it is having dense cytoplasm and prominent nucleus. Now, the significance of pollen grains. Pollens are harmful. Many plant species, their pollens are harmful. They cause allergy. So many species, they are allergic. Their pollens are allergic. They leads to chronic respiratory disorders like asthma and bronchitis. One example of the plant whose pollen causes allergy is Parthenium or carrot grass. This came to India as contaminant with imported wheat. This is Parthenium. And you can see this is the Parthenium dermatitis caused by the pollen grains of carrot grass. Pollen grains, they are rich in nutrients also. So in the market, you will find pollen tablets and syrups which are used as food supplements. They are given to athletes and racehorses to improve their performances. Coming to viability of pollen grain. Viability of pollen grain means once they are shed, okay, till what time they will be able to germinate on the stigma. Okay, so viability of pollen grain, it depends upon two factors. One is temperature and other is humidity. And the viability of different pollens is different. For example, the pollens of rice and wheat, their viability period is 30 minutes. Means once they are released within 30 minutes, if they lands on the stigma, they are going to germinate. Otherwise, they won't be able to germinate. In some members of Rosaceae, Leguminosae and Solaminaceae, their pollens are viable for months, 1 month, 2 months or 3 months. Now, pollen grains, we can store them for years in liquid nitrogen at minus 196 degrees Celsius. This is 196 degrees Celsius. Okay. So, at this low temperature, all the metabolic activity stops and we can preserve them for several years. And the pollens of good quality, they can be used in crop breeding programs for crop improvement. Okay, like we have seed bank, we have sperm banks. Same way, pollen banks are also there where we preserve the pollen grains in liquid nitrogen. So that's all regarding the androecium, the pre-reproductive structure, the male reproductive part. Now, these are few questions which are asked in board exams. So go through the questions and find out their answers. If you have any doubt, you can ask in the comment box.